Hello and welcome back. And this will be for the last time. So this will be our last video in the series for Biology 142. And this particular topic is a really interesting topic. It's human development. It is amazing how humans develop from single cells into the trillions of cells and all the complex organs that make up all the systems of our body. So this process begins at fertilization. Now, there are things that sperm cells have to overcome in order for the sperm cells to fertilize an egg cell. And just physically, millions of sperm cells are going to leak out of the vagina after sexual intercourse and never have an opportunity to swim towards the egg cells. So there is a vaginal acid that's created, and that acidity normally helps to inhibit microbial growth, but it also will destroy sperm cells. And sperm cells are swimming in a fluid that's alkaline, but not all of them are going to, be protected by the alkaline fluid, and some of them will die from acidity. So, of course, lots of them will fail to make it to the cervix, which is the entrance to the uterus, and some of them are actually destroyed by phagocytes in the uterus if they do make it to the uterus. Before they can fertilize an egg cell, the sperm cells have to be what we call capacitated. That is, they have to have these surface proteins and lipids kind of rubbed off of the acrosome membrane, so that that membrane will be able to be activated later on. So they do have to swim, and they have to knock into the walls of the female reproductive tract to help capacitate and wear down the membrane around the acrosome. So about 1,000 to 200 are going to make it to the uterine tubes when it's all said and done. So the events to fertilization, one of the first things that the sperm cells have to do when they arrive at the egg cell is they have to digest through the corona radiata. Those are granulosa cells that are left over from the when ovulation occurred. That corona radiata is going to be digested by surface enzymes that are on the surface of the acrosome. So once the sperm cells make it through the corona radiata, they're going to bore a little tunnel through. The sperm cell receptors are going to join with the surface receptors on the zona pellucida. That's that protein layer on the surface of the egg cell. This is going to cause the acrosome reaction to occur, where that little bag of digestive enzymes on the surface of the head of the sperm cell, they're going to open up, and digestive enzymes are going to be dumped onto the zona pellucida. So a little tunnel is going to be formed, and one sperm cell will eventually reach inside of the zona pellucida and reach the receptors, the little protein receptors that are going to be on the oocyte. And that's going to cause them to fuse with the oocyte membrane. Once the sperm attaches to the receptor of the oocyte, it causes calcium stored in the endoplasmic reticulum of the oocyte to be released. This calcium is going to do several things. It's going to cause meiosis II in the oocyte to be completed, and it initiates the cortical reaction. So enzymes, this cortical reaction will cause enzymes that are stored inside the oocyte to be dumped into the extracellular space, and they're going to digest all the other sperm cell receptors on other sperm cells. They're also going to cause fluid to enter in underneath of the zona pellucida to fill that space and kind of push the zona pellucida away from the oocyte, and to push the corona radiata away, thus pushing sperm cells away from further fertilizing the egg cell. And this cortical reaction is really important because it prevents polyspermy, which is the fertilization of the egg by more than one sperm cell. Okay, you don't want extra genetic material to be in there, so we have to have a reaction that gets rid of that. So as you can see in this diagram, there is a path that sperm have to follow. They have to go through the corona radiata, they have to go to the zona pellucida, they then have to get to the plasma membrane of the oocyte, and then eventually they're going to make it into the oocyte. So you can see the sperm cells kind of digesting their way through, and eventually here's a little path that they created. One eventually slides through and enters in. And this is just showing you kind of like, the sperm cells are kind of going in. Multiple ones of them are having the acrosome reaction, and then eventually one of them will join with the receptors on the oocyte and actually penetrate on through. But it does take multiple sperm to drill or to penetrate a hole or a pathway through, using all the digestive enzymes inside of them. So that haploid, which means having one single set of chromosomes, that haploid nucleus is going to become the male pronucleus once it enters into the oocyte. The haploid nucleus of the fertilized ovum becomes the female pronucleus. And when these two merge, it's called syngamy. When these two merge, they form that diploid zygote that will become a new human being. And here's what the pronuclei look like inside of an egg cell. Now, depending upon what book you read, some books will have that there is a pre-embryonic period, and that extends from the zygote up to where the cells will enter into the uterus and start to implant. But then other books will include the embryonic period as extending from fertilization all the way through the 8 weeks of development. So it just depends on which book you're reading, I guess. So after fertilization, at about 24 hours, the zygote begins mitotic divisions. These are regular cell divisions, and it's called cleavage in this particular sequence. The first division takes about 6 hours, and then successive divisions take less and less time. So you're taking and splitting that large oocyte now into two. Here's one, here's two cells during the first cleavage. Those cells are really large. They're not as big as the oocyte because they just split the oocyte in half. But they're going to become what we call blastomeres. Notice that the zona pellucida is still intact, although it's pushed away a little bit, and there are polar bodies there that will be eventually disintegrate and be removed. So by the second day after fertilization, a second cleavage is completed, yielding 4 cells. So what's kind of cool is that in an oocyte, there are chemicals that are distributed inside the oocyte in unequal proportions. So when you begin to divide that oocyte into multiple cells, you get different amounts of these little cytoplasmic chemicals in here. So as you keep on dividing, notice that the cytoplasmic chemicals up here are different in number than they are down here. And these will be signals that will help the cells to understand or to know where they belong, in what position, and what thing they're going to form in the future. So as cells become smaller and smaller, they get less of these cytoplasmic determinants, and that again helps us to understand orientation and what the cells need to become. By the end of the third day, there are 16 cells. Each of these divisions yields smaller and smaller and smaller cells. Each of the cells are still called blastomeres. So by the fourth day, the cluster of cells resembles a mulberry, which is a little berry. We have mulberry trees around here, and we call it a morula. And it's still surrounded by the zona pellucida, and it's still the size of the original zygote. This just shows you a sequence of kind of where things are going. We have here we had the female pronucleus, male pronucleus. They're all fused together to form the zygote. Here we have the first cell division, second cell divisions. Then we have the morula that's formed. Eventually, we're going to get down here. We'll actually form a little embryo and get down here and implant it in the uterus. Notice that the site of fertilization is up in the uterine tube. The site of implantation, though, is down here in the wall of the uterus. So on day 4 or 5, the morula enters the uterine cavity. It's going to be nourished by all those glands in the uterus, and we call that uterine milk. It's a glycogen, which is sugary rich solution from all those endometrial glands. There's also stored nutrients as well in the cytoplasm of the cells that they can use as well. So at the 32 cell stage, the fluid now inside the morula is going to rearrange the blastomeres into a large fluid-filled blastocyst cavity. It's also called the blastocele. So we're going to form a hollow cavity inside of this little ball of cells by taking some of that uterine milk and pumping it in and using that as the fluid. The mass now is going to be called a blastocyst. So it moves away from being the morula to the blastocyst. And again, it's still about the same size as the zygote. And this is what a blastocyst looks like from the outside. If you take and do a cut view of it, it looks like this in a cut view, where you can see the blastocyst cavity or the blastocele. So as the embryoblast forms, two different cell populations are going to arise. We have the embryoblast, which is the inner cell mass, which will become the embryo. And then we have the trophoblast, which is the outer cell mass, and that will develop into various kinds of membranes, specifically the chorionic membrane or chorion, that will surround the fetus and make up the fetal portion of the placenta. So it's kind of cool that you have cells. You have basically genetic instructions to build an embryo, but the zygote also had instructions in order to build membranes that surround the growing baby and to form what we call the placenta, which will be the way that the baby connects to the mother. So here you can see the embryoblast, the inner cell mass. So it basically is this material right here. That's what's going to become the embryo. All these other cells right here will essentially form membranes around the baby and then form the placenta that will connect to the mother. Now it always comes up, well, how are twins formed? I always get that question when we talk about embryology. So twins can form in many different kinds of ways. So here you can see that the blastomeres separate as opposed to being joined together like in regular development that occurs. If each blastomere is separated, it can become an inner cell mass and become an individual baby. So here we have the inner cell mass is formed, but the inner cell mass actually splits, and if it splits, you can form two separate human beings. If the inner cell mass doesn't split, you can form Siamese twins. So wherever the inner cell mass did not disconnect, that's where you'd have a connection between the babies. Now there are different kinds of twins. We have dizygotic twins and monozygotic twins. Dizygotic twins are where develop when a female ovulates more than one egg, and so each of those eggs is fertilized separately by a sperm cell, and then they develop into individual humans. Now sometimes some different things can happen. The egg cells can implant separately, or they can, in this example right here, and they form different placentas, so the baby's in their own different placenta. So sometimes if we get to, in the monozygotic twins, you only have one egg cell that, of course, sometimes the inner cell mass will separate. Babies will be in the same, share the same placenta. Sometimes the inner cell mass will separate a little bit later, and you can be inside the same amniotic cavity. Sometimes they will do just all different kinds of things. Okay, so there are different kinds of twins. We have dizygotic twins, where you have separate sperm cells and egg cells that join that fertilize. Monozygotic twins come from one sperm cell, one egg cell. But you can have babies that share the same chorion, the same amnion. They can share the same placenta, or they can have separate placentas. So it all depends on how the development occurs. So the blastocyst remains free in the uterine cavity for about 2 days. Then it's going to implant Okay, so it's going to drill through the endometrium using digestive enzymes, and it's going to enter in and completely then be surrounded by the endometrium of the uterus. Implantation usually occurs in either the posterior portion of the fundus or the body of the uterus. The inner cell, and if it doesn't happen in those locations, it can become problematic. The inner cell mass will orient towards the endometrium, as you can see right here. The inner cell mass is oriented itself towards the endometrium. Okay, so here we can still see the blastocele, the cavity out here. We have the trophoblasts surrounding around. Here's the inner wall of the endometrium that will have to be digested through. Now there are endometrial glands that are supplying that uterine milk. Here is an exit of that gland, so it exits out through that little pore there. The endometrium is vascular rich, lots and lots of blood vessels that will play a pretty important role in just a little bit, we'll see. So after implantation, the endometrium is called the decidua. Decidua means shed, so part of it will shed when the baby is born. So it will shed from the it'll be the portion of the endometrium that will shed when the fetus is delivered. There are different regions of the decidua, and they have different jobs, and those are named relative to where the blastocyst is implanted. And this just shows you decidua basilis, decidua capsularis, and decidua parolitis. So those are in different regions depending upon where they're located. And just to kind of keep you up to speed as to what's happening, we had ovulation, we had fertilization, then we had cleavage, making smaller and smaller cells, the morula, the blastocyst, and then we had implantation. This occurs 6 days after fertilization. Okay. So about 8 days after implantation, the trophoblast develops into several different structures, the syncytiotrophoblast and the cytotrophoblast. Around 8 days, the embryoblast also develops into 2 layers, the hypoblast and the epiblast. And what you'll see pretty soon is that essentially embryos are going to be made of 3 layers of cells. They're going to be made of endoderm, they're going to be made of ectoderm, and they're going to be made of mesoderm. The endoderm, ectoderm, and mesoderm will all become the organs that make up the human body. So from 3 layers of cells, all organs in the human body are made. And there are certain organs made by the ectoderm, like brain tissue and skin. Certain organs are made from the endoderm, like all the inner linings of your digestive organs. And then there are certain parts that are made by the mesoderm, which would be things like connective tissue and muscle. So it's all really, really well organized. So cells of these structures form a flat disc. So cells of these, the hypoblast and epiblast, are going to form a flat disc called the bilaminar embryonic disc. Bilaminar means 2 layers, and it forms a little disc. So the amniotic cavity will forms from the epiblast, as you'll see in just a second. And the amniotic cavity is a really important cavity because it surrounds the baby and puts it into surrounds it with a membrane that has a fluid inside that the baby swims and floats in. Okay, so kind of looking at a few things here, we have the hypoblast and the epiblast. The hypoblast is this yellow sequence of cells here. The epiblast is this sequence of blue cells here. If you notice, there are cells of the epiblast that move around, and they're going to form the amnion, which is the membrane that will cover over the baby, and then there will be the amniotic cavity, which will be the fluid-filled cavity the baby will be swimming in. Now, if you ever heard of a woman breaking her water, that's when the amnion breaks and the amniotic fluid rushes out through the cervix and out of the vaginal canal. Once that occurs, it's the baby's going to have to be born because there's no longer now a barrier for microorganisms to resist microorganisms coming in. So if you kind of look at some of these other structures, we have, you can see here that there are membranes that are forming, and you can see cells are migrating and forming a layer of cells all the way around, as you'll see in a little bit. So here we can see the cytotrophoblast, and you can see the syncytiotrophoblast. Okay, so these will be basically structures that will cover around the whole baby, and then structures that are going to be forming or helping to form the placenta. So the amnion roof forms the roof of the amniotic cavity. Eventually it surrounds the entire embryo, as I showed you just a second ago, and it fills with amniotic fluid. Also on the 8th day, there is a yolk sac that will form. The yolk sac is kind of cool. In a chicken egg, the yolk sac is stored nutrients that feeds the chicken as it goes through development, because there's no placenta because they're inside of an egg. Your yolk sac, one of its jobs is going to form blood vessels pretty early on. On the 9th day, there are going to be small spaces called lacunae that will form. And by the 12th day, they fuse to form lacunar networks. So here's those lacunae that are forming from the syncytiotrophoblast, and these will be little chambers that are going to eventually fill up with the uterine secretions and then eventually blood cells from blood vessels. Okay, so you can still see the cytotrophoblast, and it's moved around all the way around. It's going to protect the baby and form a structure around the baby. Here you can see the amnion is formed, and the amniotic cavity is inside here. And here you can see that that these cells here have migrated around, and they formed the yolk sac, which will be a really important location for blood cell development. And we'll see that in a little while. All right, so this was a frontal section through about 9 days after fertilization. This is a frontal section at about 12 days after fertilization. So you can see the amnion and the amniotic cavity have gotten a little bit more developed. We have a lot of these lacunae with maternal blood vessels opening up into these lacunae. So they're filling up and pulling with blood. That will be really important when we build the placenta in just a little bit. You can also see that we have mesoderm that's beginning to form and spread out, and I'll talk about how that forms in just one second. So at about the 12th day after fertilization, the extraembryonic mesoderm develops, and the cells form a connective tissue layer around the amnion and the yolk sac. Large cavities develop that fuse and form the extraembryonic coelom, which is a hollow space. The extraembryonic mesoderm together with the trophoblast form the chorion, which is a covering around the whole embryo. The chorion is a really important structure. Let me just back up and kind of show that to you where it's located. So these extraembryonic mesoderm cells, they're going to surround all the way around. You can see that the cytotrophoblast has already formed a structure around there. So between these two structures, we now have a nice covering all around the whole, it'll cover around the whole fetus, and it'll form the chorion. So the chorion is really important because it blocks antibody production by the mother. So it will promote the production of T lymphocytes that suppress the immune response in the uterus, so that the baby won't be attacked by various kinds of T cells. Also, the chorion is going to be important too because it produces chorionic gonadotropin, or human chorionic gonadotropin, HCG. This hormone will mimic luteinizing hormone, and it will help maintain the corpus luteum. The corpus luteum is going to produce the progesterone and the estrogens that prevent the menstrual cycle from occurring, or menses from occurring or shedding. So if this chorion doesn't do its job and produce human chorionic gonadotropin, then the baby will be lost when the menstrual cycle begins after the 28 days. So if we take a look at what's occurring here, very early on, you can see that human chorionic gonadotropin is being produced in huge amounts by that chorion, and that will help to sustain the corpus luteum. Later on, as the placenta develops, the placenta becomes an endocrine organ and makes estrogens and progesterones, and you can see that the amount of those go up significantly and will help sustain the pregnancy and not allow the menstrual cycle to shed and lose that the inner lining. So I always thought it's cool to look at a chicken egg. I love chicken eggs, and you can see this is an egg. It has a shell there. But I wanted to tell you that humans aren't much different than chickens in the sense that we have an amnion, we have a yolk sac. Of course, we are embryos. We'll see that later on an allantois is going to be formed. A chorion is formed. So we have all of the structures that a chicken egg has. But we have some extra structures such as the placenta that allows us to be mammals and not chickens. So the first major event at the 3rd week of development is what we call gastrulation. The two-layered embryonic disc will transform into a trilameter. That's a 3-layered embryonic disc, and it'll contain ectoderm, mesoderm, and endoderm. Remember, those are the 3 layers of tissues that are going to become all of the organs in the human body. Gastrulation is associated with the rearrangement and migration of cells from that epiblast. The first step in gastrulation is the formation of what we call a primitive streak. So if you looked at an embryo, this is an embryo here, and we can see that the epiblast cells there in blue. You can see the yolk sac in yellow being formed. So here was the epiblast, here was the hypoblast. The epiblast is going to form those 3 cell layers. In this particular diagram, it's still a bilaminar embryonic disc. Only 2 cell layers can be seen. Okay, and we have the primitive streak there, which is a little groove that forms, and we'll see what that does in just a few minutes. We can see the amnion, the covering around that's going to cover around the baby. The primitive streak establishes the head and tail ends of the embryo. Next, there are going to be cells of the epiblast that will move inward from below the primitive streak and undergo invagination. Following this, 3 germ layers will form. So here we can see the endoderm, and then we have cells that are migrating in through the primitive streak. They're going to form the mesoderm, and then we have the ectoderm, and that makes up the trilameter embryonic disc. Okay, which is pretty amazing that all of this occurs. About 16 days after fertilization, the notochordal process will form. By about 22 to 24 days, this process becomes a solid cylinder of tissue called the notochord. Notochord is important because it's going to dictate where different parts of your nervous system are going to be forming. There is this process called induction where cells will tell cells over top of it or underneath of it what they need to become and to stimulate them to develop into the right kind of structure or tissue. The notochord will induce the development of vertebral bodies and some parts of the vertebral discs. So this is basically just showing you a dorsal and section view of the trilameter disc. So you can see the trilameter disc there with the ectoderm, the mesoderm, and the endoderm, which will make up all the layers of the baby. Oh, and there's that notochordal process that's beginning to form right there. This is the oropharyngeal membrane, so this is where the mouth is going to form down here. So we're starting to get, indications of where the head and tail are going to be. Here we have the cloacal membrane where the tail end is going to be. So we already have headness and tailness beginning to form. This is a sagittal section through the trilameter disc to show you the ectoderm, the mesoderm, and the endoderm. Amniotic cavity there, just to kind of show you orientation. Here's the head end down here, and here's the tail end down here. Here we have the allantois that's developed, which is an extra membrane that will store some waste products as the embryo is developing. And if you notice, there's a connecting stalk that's beginning to form as well. Also, during the 3rd week of development, you're going to basically form the oropharyngeal membrane. That's where the mouth is going to be eventually formed. Cloacal membrane, which is kind of the tail end, and then the allantois, as I just showed you, the membrane that stores some waste products as we develop. So that notochord that you can see right here, that little rod or cylinder of tissue, is going to induce the development of the neural plate. The neural plate will be where the nervous system is going to develop, and that's pretty important. The plate develops into a fold, so you can see a fold occurring right there, and it's going to form a groove. The lateral edges are going to become elevated. As you can see, they're elevating in this direction here. They keep on elevating and migrating towards each other. And eventually they seal off and form a tube called the neural tube. That neural tube will become your spinal cord and your brain, and it will fold to form different folds of the brain. And this is called neurulation, and a really important part of development. If you look up here, here's a dorsal view of it. So here you can see where it's actually fused together, and hasn't quite fused together up here or down here yet. Notice too that the embryo is now bending this way towards the sides, and it will also bend in these directions as well to give three-dimensional shape to it. As the neural tube forms, some of the ectodermal cells will form the some of the ectodermal cells from the tube migrate and are going to form what we call the neural crest. So here we can see neurulation in process. Here we start with the neural plate, neural folds, then we have the neural tube. There is a neural crest, and these are going to form, cells that are going to go out and migrate to form nerve cells that migrate around. There's that notochord, which was important in forming that neural tube. At about 4 weeks after fertilization, the head end of the neural tube develops into 3 enlarged areas called the primary brain vesicles. So we have the prosencephalon, or the forebrain, the mesencephalon, or the midbrain, and the rhinencephalon, or the hindbrain. And these are going to form things like the cerebral cortex, the midbrain, and then the things like the pons and the medulla. So you can see they fold, so these vesicles are going to be folding, and they fold and make different sections of the brain. So you can see it's the different differentiation, or different parts of the brain are beginning to form. At about the 17th day after fertilization, paired cube-shaped structures called somites will form. Here's your somites right here, and we're going to have 42 to 44 pairs that will be present. Each somite will develop into a myotome, a dermatotome, and a sclerotome, and these will form various bones and muscles in your body. And you can see they're beautifully arranged here in this embryo picture. You can see kind of like the folding of the brain up here into different sections, which is pretty cool. And it even has limb buds that you can see right here and here. At the beginning of the 3rd week, the formation of blood vessels, which is called angiogenesis, occurs, and you get the development of blood islands. So these blood islands are going to, as you can see right here, they will form, and we get blood cell formation that will occur, and eventually these little blood islands and lacunae that they make eventually form blood vessels. And this occurs in the wall of the yolk sac. On days 18 and 19, the heart begins to develop into the in the head end of the embryo. It begins in a region of the mesodermal cells called the cardiogenic area. So the heart actually forms from a pair of endocardial tubes. These tubes will fuse together to form a primitive heart tube. This is a pretty amazing area as well. So 19 days, we have cardiogenic regions. In these cardiogenic regions will form these little tubes. So you have a pair of tubes. You can see blood moving in these directions here. Here they form, they basically will fuse together to form one tube. That's at 22 days. And that tube will begin to fold. So literally the tube bends and folds. So at day 24 you can see it's folding. This little portion right here will go up this way and form the atria. And of course this portion here forms the ventricles. So basically it folds and folds. This is showing you the same thing, I guess. The tubes fuse together to form one tube. They then begin to rotate and fold, thus leading to the formation of the final heart, which is right here in this beautiful picture of an embryo. Embryonic tissue is going to eventually invade the uterine wall and erodes the uterine blood vessels. Blood fills those lacunae. So you can see the lacunae that have formed. Once the blood vessels of the mother have been eroded, they will fill up those lacunae. By the end of the second week, the chorionic villi are going to develop. These will be basically extensions out of the baby portion of the placenta that will help the baby to feed on maternal the fluids inside of maternal blood. By the end of the third week, blood vessels develop in the chorionic villi. You can see blood vessels developing in these little villi. So yeah, villi, increased surface area is what you're thinking about. They are going to connect to the embryonic heart. Down here, the connecting stalk will go through and connect to the embryonic heart. So, so here you see these lacunal spaces that are going to be filled with blood. Mother's blood cells don't go into baby circulation. There are tissues that divide those things. But the nourishment from mother's blood will go into those blood vessels and go back to the embryo to nourish the embryo. The vessels connecting to the heart do so by way of the umbilical arteries and the umbilical vein through the body stalk, which they eventually become the umbilical cord. So here you can see the umbilical veins, and we can see the umbilical arteries. And we can see the spaces that maternal blood will be filling up. Here's a connecting stalk to the embryo. And you can see the chorionic villi that are forming there that will help to nourish and feed the baby. Placentation is the process of forming the placenta. This structure is the site of exchange of nutrients and waste between the mother and the fetus. The placenta produces hormones used to sustain pregnancy. So that placenta, again, keeps the pregnancy from being shed because a menstrual cycle forms. So by the beginning of the 12th week, the placenta has two parts. It has the chorionic villi, the fetal portion, and then has the maternal portion made of the decidua bacillus of the endometrium. This is kind of what it looks like. So here you can see a little bit more advanced baby forming. You can see the umbilical cord. And you can see the little chorionic villi that are making up the fetal portion of the placenta. So, so in this particular portion, we have the chorionic villi of the fetal portion, and then we have the decidua bacillus of the uterus making up the mother's portion. Of course, mother's portion leaks blood into the lacunae, and baby's portion, the chorionic villi, collect nutrients from that blood and then take it back to the baby through the umbilical vein, and then new blood comes in from the baby from the umbilical arteries. It's kind of what a placenta looks like. It's been false colorized with latex to show you the umbilical arteries and the umbilical veins. And that's what the umbilical cord looks like. It's a pretty substantial cord. It feels very similar to if you've ever touched an extension cord. That's kind of what the umbilical cord kind of feels like. It's shaped kind of like a pancake. It's able to protect the fetus from microorganisms, the placenta does, but it also has the nourishing functions as well. Again, mom's blood doesn't go into baby's circulation, and baby's blood doesn't go into mom's circulation. So they are separated by those chorionic villi. And this is showing you kind of a little bit more. Here's the little lacunae filling up with mom's blood. But remember, it doesn't go into baby's circulation because the chorionic villi protect it from doing that. So all major organs develop between the 4th and 8th weeks, and this is called organogenesis. That's the development of the organs. The embryonic folding occurs during the 4th week, and this involves the flat embryo folding into a three-dimensional cylinder. So you have folds here and here at either end of the embryo, and it just gets more exaggerated as time goes on. So we have a head fold and a tail fold to develop. We then get, from the side view, we get lateral folds that form as well. And they basically curl towards the midline. They incorporate the yolk sac into the embryo, and that's kind of what you're seeing here, is eventually they're going to incorporate the yolk sac in. As you can see, they're incorporating all of that yolk sac. On the outside of the embryo is a cavity in the tail called the proctodeum. So we have the proctodeum right here, which will form part of the digestive tract. Separating the cloaca from the proctodeum is the cloacal membrane. So there's a little membrane there that does separate those particular structures. So this makes an embryo kind of look funny. It does have these things called pharyngeal arches. These are also known as branchial arches, and they develop on the sides of the head and neck during the 4th week of development. Each arch is separated by a pharyngeal cleft or space or a little groove. So parts of your neck and throat are going to be made from these particular structures. By the middle of the 4th week, the upper limb buds develop. So the upper limb buds develop. By the end of the 4th week, the lower buds are going to form your leg buds. By the end of the 4th week, the embryo has a tail as well. During the 5th week, the brain and head develop rapidly, and the limbs develop further. You can see they almost look like little paddles. By the end of the 7th week, the regions of the upper limbs have become distinct digits. By the end of the 8th week, you get eyelids that come together, and the tail disappears. External genitals begin to differentiate, and digits are distinct and are no longer webbed. Okay, I think we'll stop there, and then we'll tackle the fetal period and then the actual birthing process in the second video. So until then, I will see you later.